The overall goal of this procedure is to generate single nucleotide polymorphism genotype data using MALDI-TOF mass spectrometry. First, use PCR to amplify the DNA templates, each of which contains a SNP. Remove the unused regular dNTPs using shrimp alkaline phosphatase. Next, add SNP extension primers to generate allele-specific amplimers. Then, measure the mass of the allele-specific amplimers using a mass spectrometer. Ultimately, calculation of the magnitude of each allele signal from the results can detect the presence of the allele for a corresponding SNP.
The main advantage of using this technique over existing methods like conventional PCR is the ability to utilize multiple markers simultaneously to reduce the amount of time spent generating data.
This method can help answer key questions in the microbiology field such as hyperdigestion level, insecticide resistance, host preference, and parasite infection rates.
The implications of this technique extend towards the diagnosis of other diseases or pathogens involving multiple markers because it can be used to assay nearly any SNP or indel.
In a 1.5 milliliter microtube, combine all the PCR primer pairs of each single nucleotide polymorphism as detailed in supplement tables 1 and 2 of the text protocol. Then, prepare a PCR cocktail as described in the manufacturer's protocol. After vortexing the tube lightly, centrifuge for 30 seconds at 2000 times